Genetic engineering is a set of techniques used to manipulate an organism's DNA. There are many different techniques in genetic engineering. The different types are: recombinant DNA technique. Recombinant DNA technique adds or replaces DNA in the organism, resulting in the recipient organism containing autologous DNA. They may be the same or different species. Recombinant DNA is possible because DNA molecules from all organisms share the same chemical structure. They differ only in the nucleotide sequence within that identical overall structure. Recombinant DNA results from artificial methods in the test tube. First, DNA is selected and the selected gene is extracted. Second, a bacterial DNA molecule plasmid is picked to the selected gene. 3. A part of the plasmid is cut to introduce the gene selected. Fourth, the gene extracted in the first phase and the plasmid of the bacterial DNA molecule chosen in the second one join together and form a recombinant DNA. 5. Recombinant DNA is introduced in the bacteria. 6. Recombinant DNA is inherited to their daughter cells. The main application of this technique in different fields are: medicine, producing more effective antibiotics and greater quantities of a specific protein such as insulin. Agriculture, improving yields with crops that are resistant to herbicides or pests, or have greater nutritional value. Livestock, producing animals that grow better, have higher resistance to adverse conditions, and can produce useful substances. Waste and pollutant disposal, turning refuse and organic waste into compost and humus, clearing up oil spills and removing heavy metals. And fuel, producing fuels from living organisms or organic waste such as biodiesel.
Polymerase chain reaction. Polymerase chain reaction is a technique used in molecular biology to amplify a single copy or a few copies of a segment of DNA across several orders of magnitude, generating thousands to millions of copies of a particular DNA sequence. Developed in 1933 by Carey Smalley, who was an employee of the Cetus Corporation and also the winner of Nobel Prize in Chemistry in 1993. It is an easy, cheap, and reliable way to repeatedly replicate and focus segments of DNA, a concept which is applicable to numerous fields in modern biology and related science. Polymerase chain reaction is probably the most widely used technique in molecular biology. This technique is used in biomolecular research, criminal forensics, and molecular archaeology. The machine. A thermal cycle, also known as polymerase chain reaction machine or thermal polymerase chain reaction recycle, is a device used in molecular biology that allows to perform the temperature cycles necessary for a DNA amplification polymerase chain reaction or for sequence reactions with the method of Zank. The process are DNA denaturation, the temperature is increased to separate the two strands of DNA, alignment, DNA fragment starts the replication process, and elongation, the polymerase enzymes create a complementary strand. These phases are repeated successfully. And the application of the polymerase chain reactions are in medicines, such as the genetic testing and the tissue typing, in scientific research, such as in the DNA sequencing and the DNA cloning, in forensic and infectious diseases, such as the tuberculosis.
Cloning means using genetic engineering to make identical copies of the original organism's cell or DNA molecule. There are two types of cloning: therapeutic application and reproductive application. They have the same process except the final phase. The process. First, an ovum and a cell are taken. In the ovum, the nucleus is removed, and in the cell, the nucleus is extracted. Now we have an ovum with a nucleus and an extracted nucleus from the cell. The extracted nucleus from the cell is inserted into the nucleus-free ovum. Cells are cultivated to reproduce in the laboratory. Now there are different final phase 3 types of cloning. Reproductive cloning. The resulting clone has the same DNA as the nucleus donor. Therapeutic cloning. Organs and tissues produced can be transplanted to the donor with no risk of rejection.